We all had studied that day on the cells, about the cells of immune system, right? About the different cells of the immune system. So besides the cells,
Yes, ma'am.
there are different organs of the immune system, okay, which are involved in our in enhancing our immunity. Now from these organs only the different cells develop. There are certain organs from which these cells develop. So we will study in details what are those organs. Okay, so just a minute. Okay, so first of all, we'll see what is this lymphoid organ. Lymphoid organs are stationed throughout the body. They are concerned with growth, development, and differentiation of lymphocytes. There are structurally and functionally diverse lymphoid organs and tissues. They are interconnected by the blood vessels and lymphatic vessels through which lymphocytes circulates. These organs are involved in specific as well as non-specific immunity. So we had studied about lymphocytes. So where does this lymphocyte come from? These lymphocytes develop from the lymphoid organs. Okay, so these lymphoid organs, they are present throughout the body, and this these organs are concerned with what? Concerned with the growth, development, and differentiation of the lymphocytes. I had told you that that the T cells, they are categorized into different functions based on their in what they are good at, based on that. So that differentiation is done in these lymphoid organs. So the lymphoid organs, they are structurally and functionally different from each other. There are various lymphoid organs and tissues, so all of them may be structurally different from each other, all of them will be functionally different from each other, but they are all interconnected. How are they interconnected? With the help of blood vessels and lymphatic vessels, and through these vessels, the blood vessels and lymphatic vessels, the lymphocytes, they circulate. Okay, and these organs are involved in both specific as well as non-specific immunity. Now the lymphoid organs can be differentiated into two types. One is primary lymphoid organs, the other is the secondary lymphoid organs. So this names primary and secondary, they have been, or these organs have been divided into primary lymphoid organ or secondary lymphoid organ based on their functions and how they help in the development of the lymphatic cells. Based upon that, they have divided these organs into two groups. So you see, this is a human figure, and here throughout the whole body, there are different parts, and in every part there is a lymphoid organ. Starting from here, the adenoids which are present here, the tonsils. We never knew that tonsils also help are one of the lymphoid organs and helps in immunity. Then thoracic duct, okay, this vein here, lymphatic duct, then thymus which is present in this area, then spleen, the Peyer's patches, large intestines, appendix, small intestine, the bone marrow, then tissue, or the lymphatic tissue. So everything which have been named here are all part of the lymphoid, part of the lymphoid, are all different kinds of lymphoid organs. Okay. So first of all, let's see what is the primary lymphoid organ. The primary lymphoid organs can also be named as central lymphoid organs. So this lymphoid organs, the primary lymphoid organs, this is the site, or this is the place where the development of immature lymphocytes take place. The lymphocytes which develop and which develop is, the development happens in this primary lymphoid organ or the centroid lymphoid organ. So what happens here in these organs? The differentiation, the proliferation and maturation of the stem cells into immunocompetent cells take place. So examples of lymphoid organs are the thymus and the bone marrow. That day I had told you about T cells. I had also told you about B cells. Then I had told you how from the stem cells the lymphoid progenitor, the myeloid progenitor cells develop. I hope you all remember these things. So these cells which develop, they are developing from this thymus and the bone marrow. And this thymus and the bone marrow, they come under the category of primary lymphoid organs. Now, first of all, let's see what is this thymus. So going back to this particular figure, where do we see the thymus? You see, the thymus is present in this area. Okay, somewhere near the, just above the heart. Okay, in the thoracic area. This is where the thymus is present. But what is this thymus? The thymus, when we saw in this diagram, we see it as a very small structure. Okay, we cannot actually distinguish how the different parts of a thymus is, or whether a thymus can have so many different parts. Now you see, this is actually how a thymus is if you macroscopically view it. Okay, when we see macroscopically, we see that the thymus have actually two parts. One part is known as the cortex, the other part is known as the medulla. The lower part is the medulla, the upper part is the cortex. So this upper part, which is the cortex, it has again, it has been differentiated into many parts. We have dividing thymocyte here. Thymocyte are the thymus cells which are dividing. So this division takes place here in the cortex area. This part of the of the cortex is known as trabecula. This area is known as capsule. Then the dead cell are also present here. These are all thymocytes. Then these are known as the nerve cell. Then and these are the epithelial cell, cortical epithelial cell. Then within the medullary area, you will find the presence of macrophage. The presence of blood vessel will be in the medullary area. Then this is the Hassel corpuscle. Then these are dendritic cells which are present, and these are the medullary epithelial cell. So you see, these two areas have been divided inside the thymus. Now let us study about the thymus. This thymus is a flat bilobed organ situated above heart. Each lobe is surrounded by a capsule. Okay, this capsule surrounds the thymus. Lobe is divided into lobule and are separated by connective tissue called trabeculae. The trabeculae is this. So these are the connective tissue which is separating the lobes of the thymus. The lobe organized into two compartments. One is the cortex. This is the cortex area, which is the outer part, and medulla is the inner part of the thymus. What is the role of thymus in immune function? Like how has this been studied? That the thymus has a role in immune function. How was this proved? So this was proved because by a study. The study was thymectomized mice. That means what? They used a mice. They studied on a mice whose thymus has been removed. When the thymus of that mice was removed, and then you know, further on studies were done on that mice, they found that the mice was uh, immune compromised. The mice had no immunity, and when it was um, when it had to face different kinds of infection, the mice was not able to protect itself by the by its own immunity, because the thymus was not present. The T cells were not being able to be formed in that mice. So that is how they studied. They also found that in certain mice there might be congenital birth defect. Congenital means when they are very small during their birth. They might have some defect. So that kind of defect is known as George syndrome. In that kind of defects, the thymus is absent. Okay, so in those cases also, they have found that those kind of mice whose thymus is already absent by birth, their immune system does not work properly. Then another point was found out that aging is accompanied by a decline in thymic function. Now the role of thymus is what, as I told you, is to develop T cells, and these T cells, they help in our immunity. They help in the formation of this immune cells. So, but what happens if a person starts aging? That means aging means the person starts becoming old. There is decline in the thymic function. So if there is decline in the thymic function, that means the thymus will not work properly. If the thymus doesn't work properly, that means there will be no formation of T cells, or there will be less formation in T cells. So this was also experimented using mice. Thymus grafting was done in mice, and then they found out that aging is accompanied by decline in thymic function. Okay, so after thymus, the next part is the bone marrow. So this is also a primary lymphoid organ, bone marrow. So what do we know about the bone marrow? Bone marrow is the site where B cell originates. The origin of B cell and development takes place in the bone marrow. Bone marrow has two regions. One is known as a red bone marrow, another one is yellow bone marrow. See here, this is the bone marrow. Hmm. Bone marrows are usually found in the long bones. Long bones are like the bones of your thigh area. That is a long bone, or the bone from your shoulder to the elbow. That's also a long bone. Okay, so those long bone, they consist the bone marrow. Now bone marrow are of two types. One is the yellow marrow, another one is the red marrow. These are the yellow marrow and this is the red marrow. Okay, then this is the nerve. The blood vessels are this, through which the blood flows, and then the compact bone, this is known as the endosteum. This part is known as the periosteum. This part is the trabeculae, and this area is known as the spongy bone. Okay, so these are the different parts of a bone marrow, I mean bone. Now let's see about the bone. The bone they have got two regions. One is the red bone marrow, another one is the yellow bone marrow. Now uh, B cell and stromal uh, B cell and stromal cell within bone marrow interact with each other and secrete various cytokines. Eliminate B cell with self-reacting antibody, and in birds these are known as bursa of Fabricus. See, just like the human beings, bone marrow is present. In birds, you don't find bone marrow. What is present in birds? The bone marrow in birds is known as bursa of Fabricus. So what does this bone marrow help in, or how does it help? The bone marrow does the production of B cell. B cell and stromal cell, which are present in the bone marrow, they interact with each other. The B cell and the stromal cell, they interact with each other, and when they interact with each other, what they do, they secrete a chemical. That chemical is known as cytokines. So various kinds of cytokines are secreted by the cells. Then what happens if when this B cells are present within the bone marrow, they will react, they will self react, sorry, the eliminating B cell with self reacting antibody. So this B cell will produce some antibodies. When they produce these antibodies, they will start reacting with the antigens. Okay, so this is what happens in the bone marrow. Then after the primary lymphoid organs, what comes next is the secondary lymphoid organs. Now, this secondary lymphoid organs are also called as the peripheral lymphoid organs. So this secondary lymphoid organ is what? It is where the antigen is localized so that it can be effectively exposed to mature lymphocytes, initiate adaptive immune response. So in order to initiate an adaptive immune response, we will have to expose the antigen to the lymphocytes, and this exposure of antigens to the lymphocyte is done within the secondary lymphoid organs. Okay, the examples of secondary lymphoid organs are lymph nodes and the spleen. So this is the lymphatic vessel where you find the presence of the lymph nodes. The lymph nodes are present here. So this lymphatic vessel is what? It is a network of vessels that collect fluid that has escaped into the tissue spaces from capillaries of circulatory system and ultimately returns to blood. Now see, just like the way how the blood flows in our body through the arteries and veins, similarly there is a fluid which is called the lymphatic fluid, which flows in our body. This lymphatic fluid, they how where through where does it flow? It doesn't flow through the veins or the arteries through which the blood flows. It does not do that. It flows through these vessels. Okay, and this vessels or this system um, through which this lymphatic fluids are flowing is known as the lymphatic system. And what happens, these vessels, they have this fluid flowing within it, and this fluid, and in ultimately it returns to the blood. It goes and get mixed with the blood. So this in this fluid or in the lymphatic system, there is something which is known as lymph nodes. Okay, there is a thing which is known as lymph nodes. I don't know whether the lymph nodes are shown here or not. Here, yeah, this is the lymph node which is shown here. This is the lymphatic vessel. This is the tissue space. These are the capillaries, lymphatic capillaries to which the lymph is flowing, and then these all are the lymphatic vessels. This is the lymphoid follicle. This is the afferent lymphatic vessel, and then this is what the lymph node. It is the lymph node which has different parts like afferent lymphatic vessel, germinal center, secondary follicle. These are different parts within the lymph node. Now this lymph node, it is where the immune response is mounted to antigen in lymph. It is encapsulated, it is shaped like a bean, and it has got different region. So we see this is the lymph node. So what is the shape like? The shape of this lymph node is like that of a bean. So here it has been enlarged and shown. So here what do you see? This is the lymph node where the parts have been differentiated. This part, this yellow part is known as the cortex. This red part is known as the paracortex, and the medulla is this green area. Okay, this is how the different parts have been shown. So we see here divided into three concentric regions: cortex, paracortex, and medulla. Now, what this cortex does, the cortex has the presence of B cells, macrophage, follicular dendritic cell. This area, this yellow area has the presence of the B cell, macrophage, and dendritic cells. This is thymus dependent area. This area is dependent upon the thymus. Okay, this area is dependent upon the thymus. Then comes the medulla. The medulla has the presence, this area has the presence of the plasma cells. Fine. That means from the B cell, what will develop into? When the B cell is activated by the T cells, it will convert into plasma cells. And then, so from this B cell here, the plasma cells which will be developed goes into this area, this medulla area, the green color. Okay, so what happens? Antigen reaches the lymph node, then there it gets trapped, the antigen gets trapped. Then after the antigen gets trapped, class 2 MHC molecules, they, what they do, they come out of that cell, and then they show this antigen to the other cell, which results what it, when the antigen is shown with the help of this class 2 MHC molecule, then the resulting, that results in activation of the T helper cell. When the T helper cell comes and gets activated and it comes and binds to that antigen, that leads to the activation of the B cell. And when this B cell gets activated, initial activation B cell takes place within the paracortex area. Okay, so this paracortex area, I told you that in this cortex, what is present? B cells are present, macrophages are present. So this, from this B cell, in this paracortex area, the activation of the B cell will happen. When after the activation of B cell happens, the plasma cells will be formed. Those plasma cells are formed in the medullary area. Okay, so B cells are then differentiated into plasma cells, and then they secrete antibodies like IgG. This is how it works. This is how it works within the what do you say, lymph nodes. Okay, then comes spleen. I hope you all have heard about this organ, spleen, before. So what is this spleen, or what is the function of the spleen? Spleen is an immune an organ which is related with immunity. Okay, so immune response is mounted to antigen in bloodstream. It is a large ovoid structure situated high left in the abdominal cavity. I'll show you the diagram in the human body where the spleen is located. So here, this is the spleen, okay, which is located in the left side. Situated high left in the abdominal cavity. It is surrounded by capsule, extend interior to form compartments. The compartments are of two types, red pulp and white pulp. So here, this is how the spleen looks like. This is how the spleen looks like. These are the different parts of it: hilum, gastric surface, splenic artery, splenic vein, renal surface, and then here, which one is which they told us red pulp and white pulp. This area is known as the red pulp, and then these things are known as altogether known as white pulp. What are included in white pulp? Primary follicle, marginal zone, then periarteriolar lymphoid sheath. This is the primary follicle, this is the marginal zone, and this is the periarterial lymphoid sheath. So these three things comes under white pulp. There are other parts also, but white pulp and red pulp are the main thing in a spleen. Now what happens in the red pulp? Red pulp comprises of the RBCs, macrophages, and few lymphocytes. Hmm. Then the extraction and removal of RBCs takes place there in this area, red pulp. Then white pulp surrounds splenic artery and forms PALS. Lymphocytes, primary follicles present in pulse rich in B cells. So you see what happens in this area, the red pulp area, what are present? These things are present. The RBC is present, macrophage and few lymphocytes are present. Okay, RBCs, macrophages and few lymphocytes are present in the red pulp area, and destruction of the RBCs also take place here. Destruction of RBCs means we know that within 120 days the RBCs get destroyed and new RBCs gets formed. So that destruction of the RBCs takes place within this red pulp only. And then what we know about this white pulp? White pulp surrounds splenic artery and form PALS. Okay, so this is the splenic artery. This is the splenic artery, and where the white pulp is present, and here it forms this one, PALS, and then lymphocytes are present here, primary follicles are present, and those things are all rich in B cells. Marginal zone means the margin. It comprises of the lymphocytes and the macrophages. So what happens in this spleen? Blood-borne antigen and lymphocytes are when they are present there in the spleen. In the marginal zone, they are trapped by interdigiting dendritic cells. Now see here. This marginal zone, which is present here, okay, in this area, the antigens get trapped because there are some dendritic cells present over here. When these dendritic cells present over the dendritic cells which are present over here, what they do, they trap the antigen which enters. Okay, so they are trapped by interdigiting dendritic cells, okay, which are present in the marginal zone, forming to PALS. Then this leads to the activation of the B and the T cells in the PALS. In the PALS itself, the B and T cells get activated. And once they get activated, what will happen? The dendritic cell, it captures the antigen, and then it again follows the same method which it followed before in the other organs. It gets combined with MHC molecule, and then the TH cells get activated, leading to activation of the B cells and primary follicles in the marginal zone. So this is how it works in the spleen. Okay, so where did we stop here? Yeah. Then after the B cell is activated, then the B cells, they rapidly divide. After which, what happens? Secondary follicles are formed, and then they form this B cells. They help in formation of the plasma cells, and after that, leading to antibody production. Okay. Next, going to the mucosal associated lymphoid tissue. This means what? Mucosal associated means we understood what are lymphoid organs, we understood what are lymphoid cells. Now this lymphoid tissues means it into certain tissues which has the presence of mucus over it. Okay, mucosal associated, that means it is associated with mucus. So such kind of organs, for example, are tonsils, appendix, pairs, patches. These are some of the example of the organs which are lymphoid organs or lymphoid tissue, but they have the presence of mucus on it. Mucosal membrane. A membrane is mucosal. Okay. So here you see uh, they have a large number of antibody producing plasma cells in those kind of organs. See here the tonsils. We all know what is a tonsil. So what does a what tonsils does? They defend against antigen coming from nasal and oral epithelial root. So this is the tongue. These are the tonsils which are present here. Okay, so this is the lymphoid tissue. This is the crypt, cross section of the palatine tonsil. And then This mm, see here, this is the pharyngeal tonsil. So what happens? This tonsils, as they enter, sorry, this antigen as they enters the tonsil through this crypt, they, they form a lot of antibody in this area, and they will be there will be the formation of dendritic cells and macrophages, and then they will all engulf it within this tonsils only and kill it within itself. They will not let it go out uh, and infect the other parts of that area of the respiratory tract. So it happens, everything happens within the tonsil itself. Similarly, the same thing happens here in this gut associated lymphoid tissue. This is the intestine, and intestine is also mucosa, made up of mucous membrane. So in the same way, uh, in the intestine, you see these are the presence of villi on it. Okay, this is the M cell, this is the lamina propria, and these are the different organs. So what will happen whenever an infection takes place? The infection will enter, this will invade this cell, which is not so easy. First of all, it's because of the mucus which is present over there. It's not so easy for those cells to invade it. But once it gets invaded, then everything, whatever action takes place within this cell itself, within this cell itself here, and then they get destroyed. I will make like explain you all in detail one by one later in a different class how this actually work. For the moment, you'll just see. So this is just a diagram through which they have shown this, how the antigen has entered here, then how after the antigen has entered the B cell, they have what do you say, got attached with this antigen, and then with the help of this MHC they have shown, and this T cell, they came and got bound to that B cell. Okay, then the B cell, they started multiplying. This is the macrophage which is present over here, and then the B cell will ultimately form antibodies, plasma cells and antibodies. So this is what is happening here. These are the plasma cells which are forming, and then so many antibodies are releasing out, and these antibodies are going outside that area, outside the um, intestinal intestinal cell. It's going out, and then if any antigen is outside, it will start killing those antigen outside the intestinal cell also. So this thing I will explain in details later on. For the moment, you will just see what are the different types of mucus associated lymphoid tissue. So this is gut associated. First one was an example of mucus associated lymphoid tissue. First one we took is tonsil. Secondly, we saw gut associated lymphoid tissue. No, in the gut, what are the associated lymphoid tissue? Then this is the tertiary lymphoid tissue. Okay, in tertiary lymphoid tissue, the example is cutaneous associated lymphoid tissue, where we see the inflammatory response. Where the skin is the anatomical barrier, there is some epithelial cells which are known as keratinocytes. These keratinocytes, they secrete some chemicals, which is cytokines, and those cytokines, they help in expressing this class 2 MHC, and it does the further processes of destroying the pathogens. Okay, so this is about what are the different types of lymphoid organs. Obviously, we are going to study in details about these different organs. So this part until now, is it clear to you all?
Yes, ma'am.
Yes, ma'am.
Yes.
It's clear, right? You can say if you have any.
Yes, ma'am.
In another class, we can explain in details about how every part is working, or especially how the mucus associated lymphoid tissue and the gut associated lymphoid tissue is working. Okay, we'll do that in a different class. Fine. Can you all just switch on your videos once? Feeling sleepy or what?